Hello, everybody, and welcome to Silence Abounds Science at Home, where we will discover home lab magic. We will demonstrate experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and molecular biology. Welcome to my channel. Hello, everybody, and welcome to another episode of Silence Abounds Science at Home. Before we get to the main experiment today, I just want to take a few seconds to reflect what's been happening in 2025. We're coming up to the end of the year, and it's been a full year where my channel has been launched. I launched it right at the beginning of January, and it's going great. I am so indebted and grateful to all my subscribers and viewers. I truly appreciate it. And I just want to talk about what's happening in 2026. I've got so many exciting projects that I'm going to be doing and creating some very exciting experiments and videos. I'm going to be doing a lot more molecular biology, biotechnology experiments, including I'm going to be doing polymerase chain reaction experiments, DNA fingerprinting. I'm even going to do a big project on CRISPR. Yes. We're going to be looking at the CRISPR technology and doing a major 5-video project on that technique. We'll be doing a lot of microscopy, looking at the beautiful world of microorganisms, protozoa, algae, and psyllids, and many more microorganisms. It's going to be very exciting and fascinating. We're going to take a look at slime molds, beautiful creatures, fascinating creatures, and we're going to be looking at those and studying the slime mold over several videos. Also, I'm going to be doing a lot of genetic experiments in conjunction with molecular biology. We're going to be studying the Drosophila, or the fruit fly. It is the most studied organism in the world of genetics, and we're going to be demonstrating some of those genetic experiments and how we can use Drosophila in the study of genetics. Of course, there'll be many other chemistry experiments. We're going to be looking at protein electrophoresis. We're going to be doing some interesting gene editing experiments, introducing that, and many more. 2026 is going to be an exciting year, packed full of varying, exciting experiments, looking at a wide range of scientific principles, concepts, and reactions. So please join me for future episodes of Silence Abounds, Science at Home, and here's to an exciting 2026. Again, thank you to everyone who has subscribed. Those who are viewing my videos but not subscribed yet, please hit the subscribe button, hit the like button. It's all free. Please leave a comment. I like to hear from you. Happy New Year to everyone, and here's to 2026. Hello, everybody, and welcome to another episode of Silence Abounds Science at Home, where we will discover home lab magic. In today's episode, we're going to delve into the world of forensic science and detecting and analyzing unknown substances. Forensic scientists are faced on a daily basis at crime scenes and in the lab where they receive substances, whether it be in a liquid form, in a powder form, and they don't know what it is. So their task as forensic scientists is to analyze the substance to determine what it is and how it relates to a crime. Today we're going to explore some qualitative tests to look at and analyze two unknown samples that were collected at a crime scene. We're going to act as the forensic scientists and screen the substances and try and deduce what they are. There are three screening tests that a forensic scientist do. They are quick, they are cheap, and they can give initial idea of what is in the substance. Later on in the laboratory, they can do more extensive analysis requiring expensive analytical equipment like a gas chromatography mass spectrometer analyzer or high performance liquid chromatography for a more definitive answer. So let's look at the three different tests that we're going to analyze our compounds with. The first is the Biuret test. And the Biuret test is a chemical test used to detect the presence of proteins. It's based on the reaction between copper ions and the peptide bonds that link the amino acids in proteins. Proteins are made up of long chains of amino acids, which are connected by the peptide bonds. And these peptide bonds contain nitrogen atoms, which have lone pairs of electrons. The copper ions can coordinate with the nitrogen atoms in the peptide bonds. And in an alkaline environment, the copper ions bind to the peptide bonds, forming a violet-colored complex. So for the Biuret test, if there's a color change and the presence of a violet-colored complex, then you know that the compound that you have is a protein. And we're going to look at that reaction along with Lugol's iodine test. Lugol's iodine test is a simple chemical test used to detect the presence of starch. The iodine molecules fit inside the helical structure of starch following a blue-black complex. Lugol's solution contains elemental iodine and potassium iodide, which increases the iodine solubility, forming triiodine. And it's the triiodine ion, which is the main species that is responsible for the color change with starch. And the last test that we're going to look at is simple acetic acid, which is a weak organic acid that reacts with compounds such as carbonates to produce carbon dioxide. The presence of a reactive compound is confirmed by the formation of gas bubbles. So what I'm going to do is I've got reaction plates that I'm going to use to carry out the reactions. And for comparison, I've got 6 standards. I've got baking powder, baking soda, I've got cornstarch, talcum powder, I've got everyday salt, and I've got gelatin. And then I have my 2 unknown samples that I have received from the crime scene that I've been asked to analyze. So what I'm going to do is just add some of the standards to the plates, including my unknowns, and then I'm going to add the chemicals to the wells and make a note of the individual reactions. And then I can compare my unknowns against my example standards that I've used, which I'll use for comparison to determine what my unknown samples are. So let's begin the test. Okay, so I'm going to add, so I've labeled my plates with the standards on top, and then each line, horizontal line, I've labeled biuret test, acetic acid test, and the iodine test. So what I'm going to do is add the compounds, the standards, just a scoopful of each compound to the wells. So I want to put each compound going down. So I will test the biuret, the acetic acid, and the iodine test for each of the compounds. So I'm going to do that now. Okay, so I've added the compounds into my plates, and so now it's time to do the reactions. And every scientist will have a laboratory notebook or worksheet, and I've created a log for me. So I'm going to record my observations as I perform the test. So the first one is going to be biuret. So I've got the biuret reagent, and it just requires a couple of drops. So I'm going to add that to each of my wells. I think just needs a couple of drops. It's coming out very well. Let me get a pipette and add it that way. And then I'm going to do the iodine. I'm going to add 2 drops of iodine reagent. And remember, we're looking for a black reaction, indicating the presence of starch. And then finally, I'm going to add the acetic acid. And I got to observe for the presence of bubbles. So baking powder, I got bubbles. Baking soda, I got bubbles. No bubbles for cornstarch. No bubbles for talcum powder. No bubbles for salt. Gelatin, no bubbles. Unknown sample 1, no bubbles. And I got bubbles for unknown sample 2. So let's look at our reaction and make a note of our observations. Okay, so let's take a final look at our results and determine what our 2 unknown substances are. So let's review the results. So the baking powder was negative for biuret, positive for acidic and iodine. Baking soda was negative for biuret, positive for acidic, negative for iodine. Cornstarch was negative for biuret, negative for acidic, and positive for iodine. Talcum powder was negative for everything, as was salt. And the gelatin was positive for biuret, negative for acetic acid and iodine. So on our unknown 1, we got a negative, and a positive for the iodine. So what we can do now is compare that pattern with our standards. So we got a negative, positive. So what compound produced the same reaction? So if we look here, cornstarch has the same reaction, negative, positive. So unknown substance 1, we can confidently say is cornstarch. And then unknown 2 was negative for biuret, positive for acidic, and negative for iodine. And the standard compound that we had that had a similar pattern was baking soda, negative, positive, negative. So it matches unknown compound 2. So we can confidently say that the unknown substance number 2 was baking soda. Well, I hope you enjoyed that experiment introducing you to forensic chemistry of unknown substances and how easy and quick it is to perform qualitative analysis when you're in the middle of a crime scene for quick rule out, rule in. Obviously, forensic scientists use very expensive, sophisticated equipment, but sometimes they need to narrow it down and do these inexpensive, quick, qualitative analysis before moving on to a more definitive confirmatory testing, which is expensive and very sophisticated, and you really don't want to be using those types of equipment for a general screening test. So please hit the subscribe button, keep supporting the channel. Thank you for my subscribers. If you're viewing this video and haven't subscribed, please go ahead and hit the subscribe button. It's free. Hit the like and share buttons. It helps support my channel. Please leave me a comment. I like to hear from you. And always remember, you can always email me with questions, comments. If you've got questions about the video, the content, and want to know more, hit me an email. And also send me your photos and videos of you doing home experiments yourself. I want to build a home lab community. Until next time, keep on experimenting. Goodbye, everybody.